There are only two cell types in the human body that can regenerate tissue and organs: stem cells and fibroblasts. The latter are the primary cells in connective tissue. And while it's understood that they collectively comprise the extracellular matrix responsible for maintaining the structural integrity of human tissue, they are perhaps an undervalued asset in the space of regenerative medicine, or at least so think the leaders of FibroBiologics. FibroBiologics is a clinical stage biopharmaceutical company with a phase 2 lead asset called CyBroCell, developed to address degenerative disc disease. But beyond disc regeneration, which might sound like an obvious job for fibroblast technology, FibroBiologics is studying the application of fibroblasts in some less evident indications, including a phase 1 study in multiple sclerosis and preclinical work in cancer immunotherapy and cancer therapeutics. I'm Matt Pillar, and on today's episode of the Business of Biotech, I'll be learning from Pete O'Hearn, Chairman, CEO, and founder at FibroBiologics, and Hamid Khoja, Ph.D., who serves as the company's Chief Scientific Officer. Prior to launching FibroBiologics, Pete was CEO of NeoSurge Technologies and Fibrogenesis, having launched his life sciences career in the late 80s with Christus Health. Dr. Khoja joined the team last summer after many, many years doing research and application science for the likes of Eli Lilly, Sequinum, Digilab Genomic Solutions, and Colvaris. So Pete and Hamid, it's great to have you guys on the show, and welcome. Thank you for joining.
Thank you. Thank you for having us.
My pleasure. Peter, I want to start with you. So back in the 90s, you left a pretty sizable health system in Christus to lead a biotech. You'd been there for a little while, and you left there to get into the biotech leadership and eventually entrepreneurial space. And that's what you've been doing since the mid 90s. It's quite a departure. So tell me a little bit about that. What prompted that move back then?
Well, I think I thought originally I'd grown up to be a hospital administrator. And when I joined Christus, I was doing product development. We built probably the first sports medicine program in the Houston area, if not the state of Texas. And working closely with doctors, I found out they are a unique group. They all are natural inventors. They all have the resources to prototype, file patents, and then they kind of stop and they don't know where to go from there. So group of surgeons talked me into leaving, and we designed a set of surgical instruments that we took to every venture capitalist in the country and told them we designed the best instrument system in the world. And they said, no, you didn't. Over 100 meetings.
Wow.
And 25 patents later, we returned 950% to our shareholders. And we were right. We went back to those VCs and we thanked them for their guidance and told them what we'd done. And most of them said they're check writers. And we said, well, we just deal with individual surgeons and physicians. But one of them called and said that they had that his brother was a French neurosurgeon and his wife was a French dermatologist. And they had this idea, provisional patent, being a neurosurgeon, said you could take a fibroblast, inject it into the spine, you could potentially regrow the disc. And everything we've worked on in the past were kind of improved technologies. And this one, if it worked, was a game changer. Mm-hmm. So we acquired that patent, came back and began work on that. And I'm proud to say we've organically written almost 400 patents to go along with that original provisional. So we've seen, we've been, I think, prolific in the lab in the last 5 years, and we've seen application for fibroblasts far outside of the disc of the original idea into almost every element of chronic disease.
Yeah. Well, you skipped ahead quite a bit there already, Pete. We'll get into some of that more recent backstory around what FibroBiologics is doing shortly. But I just want to comment, I guess. You proved that if a group of surgeons, if you work for a hospital network and a group of surgeons ask you to leave, it's not necessarily a bad thing.
Well, I will tell you that I had it was a Catholic group that owned the system, and I told them I might be back in 6 months if it doesn't work out.
Yeah.
So I said, give me 6 months and I might be back, but. That's cool. I love the entrepreneurial side. I love the kind of the blank slate, working with guys to bring something to the finish line. So just it actually became my entire career calling.
Was that something that you felt like was inside of you, even when you were working in the comfortable confines of the hospital network, that you were just kind of itching to release, and that opportunity gave it to you?
I think it was. The first 2 weeks I was there, I was just out of grad school, and I was telling was a group of wonderful nuns that ran this huge hospital system. And I said, we should buy this hospital and we should buy this professional office building. And one of the nuns said, you outgrew us the first week you were here. we had no idea that there was this. Well, and they wanted to go outside of the Catholic order and recruit in professional executives, because that Catholic order was aging and they weren't really replenishing their novitiates. And so they were looking to people like myself to come in and lead them to the next, the next evolution of healthcare. And I don't think they were prepared for someone to come in and have so many ideas. So I think that, they weren't surprised when I left with a group of surgeons. I'll say that. And the surgeons still stayed there. So we all stayed very good friends through the process.
Yeah, very good. Very good. We'll learn from Dr. Khoja here shortly whether you're still bringing that overzealous attitude to the company that you had now. We'll see how much he's willing to share. He's only been with the company since August 2021. Dr.
Yes.
Khoja?
Yeah, I joined in 2021 August, and so far it's been quite a very nice ride. I think both Pete and I are very energetic. And we love to tinker. We love to pass around ideas and test them. So it's been a great synergy between us. So we've done great so far.
Very good. Before I ask you another question, I want to make sure I Americanize everything, and worse than that, I Pennsylvania Americanize everything. So pronounce for us your last name so that I'm
no, Khoja. That's perfect. I think we chose the spelling in English that would be the easiest to pronounce. So it's you're pronouncing it perfectly.
Very good. I'll pat myself on the back. So you earned your Ph.D. in molecular biology at Boston University, and after you did that, you spent a few years in big pharma. You worked for a couple of big companies. What appealed to you about making the move to a younger, emerging biotech?
Yeah, I think it was, although I loved working at big pharmaceuticals because you have unlimited resources in terms of not only being able to do certain research, but also intellectual resources from the people, and very intelligent people that are there. But unfortunately, bureaucracy does take a toll on large pharmaceutical companies, which is the reason why they buy smaller biotechs to enhance their product lines. So for me, moving into smaller startups was, I think, a way to make more of an improvement in some of the research that I was thinking that was important. And I think that's why I moved to a smaller companies. I think this one is the fourth startup that I've been in. And I think I not only like the pace, but I also like the flexibility. Also, there's a certain sense of thinking outside the box, which is very important, I think, in scientific discovery, that I only tend to see in startups and larger companies. That tends to get dampened, unfortunately, which is natural progression. But I think that's one of the reasons why I'm very interested in being part of startups.
Yeah, very good. So Pete, you started to get into the genesis story of FibroBiologics there just a few minutes ago, and I want to dig into that a little bit more deeply. First of all, how long's the company been? When was the company founded? How long has it been around?
Since 2008-ish.
Mm-hmm. Okay. And leading into that, I mean, this was a space, if I'm reading your work history, your CEO history accurately, this was a space that you were already plugged into, or you would
well, only in the sense that we were working closely with surgeons. So the previous project was predominantly general surgeons, and this project has predominantly been neurosurgeons.
Mm-hmm. Okay. All right. So let's start at sort of a fundamental level, as I noted in my preamble, fibroblasts, obviously we know that they're a cell, right? Tell us more about it. Like, tell us more about what a fibroblast is and what FibroBiologics is doing with them.
Well, I can tell you what FibroBiologics is doing, and I can tell you what a fibroblast is, but I might be caught without a Ph.D., so I'll let Hamid give you that.
Yeah, I'm just gonna. Yeah, right. I'm gonna toss questions out there, and you guys can arm wrestle over who's gonna.
Hamid can give you the actual.
Fibroblasts certainly are one of the most predominant cells in the human body. They are responsible for the formation of connective tissue, the extracellular matrix. So they're everywhere in every organ, and they comprise multiple levels of interaction with other cell types. And other organs and other systems in the human body. What you mentioned earlier in your introduction was fibroblasts are only one of the two types of cells that can differentiate into different cell types. For example, chondrocytes, osteoblasts, epithelial cells, etc. But they also have a very predominant or prominent immune modulation capability. They do secrete a lot of cytokines, and so they can interact with the immune system very closely, as we've seen with some of the publications that have been out in the last 20 years or so. And they do have one of the reasons why we like the fibroblasts, and Pete will certainly agree with me there, is that they're so easy to source and they are very easy to grow. As compared to stem cells, which have been in the news and in research for a very long time, fibroblasts are easier to source, easier to culture, cheaper. So we find that they are much better source for some of the chronic diseases that we're studying for application. So I think it does serve as well in terms of thinking about fibroblasts as not as a replacement for stem cells, but perhaps as another way of thinking for certain diseases of using fibroblasts instead of stem cells.
We like to say that fibroblasts can become anything you need them to become, including a stem cell. If you need a stem cell, you can make one from a fibroblast. And they appear, at least in our studies in the lab, to be more potent than stem cells, much, as Hamid mentioned, much less expensive to harvest, to grow, to ship. They're just a more robust cell. And, there's a reason why we evolved with them being the most populated cell in the human body. They have an interaction in almost everything that we've studied.
Yeah.
I mean, one thing to notice, like compared to stem cells, they outnumber stem cells about 10 to 15,000 to 1. So as you can imagine, in terms of sourcing and culturing, even doubling times are significantly lower than stem cells. So you can easily culture them very much better, much more easily, cheaply than stem cells.
Yeah. Okay. All right. So I'm gonna play the skeptic here and just ask you some probably naive and basic questions to challenge the notion, right? So don't be offended. Don't be.
Oh, no, not at all. We're in science. We don't get offended easily.
Nope.
So first of all, why? I've since I've been covering this space, I've been in the space for just a few years now. Embryonic, tissue-specific progenitor, mesenchymal, umbilical cord, bone marrow, and induced pluripotent stem cells have been the conversations that I've had with cell therapy companies. Why am I not familiar with a therapeutic approach using fibroblasts?
I think one of the main reasons for that being is that stem cells have been since the patent that mentioned the use of stem cells for possible potential therapeutic uses, that has been the main cell type to mention, main cell type to research. And I think that it sort of developed into a huge market for stem cell based therapy companies, but also in research and the companies, pharmaceutical companies, small companies, even research institutions thinking of stem cells as a way of developing therapies. But I think being, and we see that in a lot of the clinical trials that have been done using stem cells, they're not that easy to work with. And I think some of these therapy companies that you mentioned that use stem cells, they would admit that, yes, they might see some clinical feasibility of them using it for a specific application or clinical outcome. Just being able to do that in a large setting for a huge number of patient sets, or in having the consistency in terms of the cells, it's very difficult. And sometimes with some stem cells and with some applications, they're practically impossible. You might be able to show it in a lab setting, in an in vitro and in vivo setting, in a small clinical study, but being able to apply that to a larger clinical therapy becomes very difficult. And that's where I think fibroblasts are superior, in that they are much easier to grow, much easier to identify, much easier to maintain and culture, and be able to provide those cells for a large clinical study or even therapeutic purpose. And I think that's the main purpose for reason why we haven't really seen a lot of stem cells coming into the clinical use. It's because of that. They're just very difficult to work with.
Yeah. So again, keeping the skeptic hat on, in science, as in life, just because it's easy or prolific doesn't necessarily make it good. So let's so let's talk about that. I mean, you mentioned that these cells can become anything a stem cell can. Therefore, like what are the applications and limitations of fibroblasts in terms of their therapeutic potential?
Yeah. Like, for example, you mentioned degenerative disc disease. So phase 2, we're looking at the data, analyzing the data to see how efficacious it has been. But in multiple sclerosis, in all our preclinical study, we compared with bone marrow and adipose-derived mesenchymal stem cells, and we found them to be significantly superior than those type of cells and immune modulation that is necessary for some of these autoimmune diseases. So we see that as well as a positive for fibroblasts. And also, if we look at other diseases, for example, some of the chronic diseases such as wound healing, right, for diabetic ulcers, etc., there are clinical studies that are looking into possibly using stem cells for those as well. But we have filed some patents, and we're starting a preclinical study on using fibroblasts for wound healing. If you look at all the publications, fibroblasts are an important part of wound healing. They are they have a role to play in every step of the wound healing, from the initial inflammation step to the tissue remodeling that occurs at the end. Fibroblasts are a major component. So we're trying to see if we could use our specific fibroblasts, dermal fibroblasts, for use in wound healing. Now, there are a lot of different types of fibroblasts. There are hundreds of subtypes. So our goal is to identify these specific subtypes in our human dermal fibroblasts and to use them for specific applications. So it's not that fibroblasts are, in general, you can use them for any application. I think that's overreaching a little bit. But I think in terms of using fibroblasts, specific subpopulations of fibroblasts for specific applications, I think that's going to be crucial. And that's our goal in the next year and a half, is to identify more subtypes for specific clinical applications.
Very good. I want to, and I do want to a bit later on get into some of the specifics around differentiation of the cells themselves for specific applications, and understand how that kind of mechanism of action works. Because as I said in the beginning, it seems to make perfect sense that if you have a cell that is structural in nature, contributes to some semblance of tissue structure, sure, let's rebuild discs with it. Let's, heal wounds with it. But its application in some of those, oncology indications, I'm curious about. So we'll get into that in a minute. But you've mentioned a few times that sourcing material is a big advantage of fibroblasts. So
yes,
walk us through that. And again, Pete, jump in here whenever you want to. So I know. Obviously, Hamid wants to talk science, and he's having a good time talking science over here. But if you want to weigh in, Pete, with like sort of the C-level CEO's business perspective on these things, that'd be great too. Because I want to know how the cells are harvested, but I also want to know, like, from your perspective, Pete, like, you got to turn around to the stakeholders and the shareholders and say, hey, what we're doing is efficient, right? What we're doing is saving us money and helping us win the race against some of these super difficult to procure cell therapies. So
yeah, and I think that what we're going to find with respect to the mechanism is critically important to us. That will reveal itself over time. But in the degenerative disc, for example, an hypoxic condition with intermittent hydrostatic pressure. So the force the vertebrae apply on that disc forces these fibroblasts to turn into chondrocytes. That's just one recipe. You're going to see it different for each of the clinical uses that we've chosen to pursue. So every one of them is going to have a little bit different, and some mechanism may be more difficult to understand than others, and that's Hamid and his team's role is to really understand that. More than just it works, people want to know why it works and how it works, and so that's important for us mechanistically, how we can explain that to the market. So I think that's important for us, and it's not always the easiest thing to determine either. You're so excited that it's working. Multiple sclerosis, for instance, it could stop the progression. Now we have to understand, how is it stopping the progression?
Right.
Yeah. And I think you asked the question about sourcing of these fibroblasts. We use excess surgical tissue from surgeries for sourcing the dermal fibroblasts. So they're very easy to source. Whereas with stem cells, you either have to do it from adipose tissue or from bone marrow, which is difficult. But also, I think I mentioned earlier, in terms of the abundance of these cells, stem cells, fibroblasts are about 10 to 15,000 times more abundant than stem cells. So you could imagine the number of cells that you can obtain from excess surgical tissue, number of fibroblast cells is quite high. Whereas with stem cells,
back up real quick. And excess surgical tissue, I need more on that. So, like, give us some examples of, like,
for example,
right?
For example, a lot of surgeries involving, let's say, tummy tuck, or any surgery in which some tissue is resected, skin tissue, can be used as a source for fibroblasts. So typically these tissues are discarded, but we have a way of using that same tissue for sourcing the fibroblasts. Okay. So and growing these fibroblasts for them to remain minimally immunogenic. So we have a way. It's not patented, but we have a proprietary way of doing that, and it works quite well. And because the fibroblast grows at much faster rate, the doubling time is much faster than stem cells. We can easily obtain hundreds of millions of cells within a matter of weeks, rather than have to deal with months and months of culturing. And also, we have to remember that stem cells are very sensitive to passaging. They tend to change over time. As you passage them more, within 9 to 10 passages, they tend to change. So you can't really expand stem cells to a great deal, because you are limited by the number of passages you can passage stem cells. Whereas with fibroblasts, you can passage them longer to 12 to 15 passages without noticing any change into the cell morphology and phenotype. So it gives us more of a freedom to operate and grow more cells more consistently than stem cells. And surgical access tissue is predominant in the US and other countries. So often they're discarded, but we can use them. And we don't need much. We only need about a few millimeters squared areas of the tissue to isolate and grow the fibroblasts.
Mm-hmm. No, it's fascinating. It's like next level recycle, reduce, reuse. I've had conversations.
Exactly.
No, for
there
real.
you go.
No patients were harmed in harvesting our cells.
Well, so I mean, all jokes aside, it's an important point. If you look at especially early days for cell therapies, a lot of the conversations around stem cells were very very contentious, very controversial, polarizing, right? And in this case, it's difficult to manufacture an argument against the responsible use of something that would otherwise be discarded. I've had conversations with cell therapy companies that do that harvest source material from placental material that would otherwise have been discarded. And it's like it's difficult for me to imagine a world in which that is not an admirable pursuit.
Right. And with certain stem cells, there's an ethical issue involved as well, right?
Sure.
So that makes it difficult. Whereas with fibroblasts, they're not there. It's not an argument.
When you're striving to excel in a new arena, the best guides are the ones already doing it well. The business of biotech brings those voices forward to help new and emerging biopharmas turn their innovations, like mRNA and cell and gene therapies, into clinical realities. Tune in and subscribe for insights on hiring, regulatory, and other need-to-know topics for biopharma leaders. The podcast is brought to you in collaboration with Cytiva. Check out their resources at cytiva.com/emerging biotech. That's CYTIVA.com/emerging biotech. So you gave us sort of a good idea of the way that the harvesting happens, collection, some insight into the process. What are the advantages? Dig into the advantages on the back end, closer to administration, for instance. I think I read, or perhaps you said something about these, the end product being perhaps hardier, more transportable. Give us some sense for that.
Yeah, I mean, because fibroblasts are very hardy, and we can culture them easily. We can remain the consistency of the product throughout the processing and manufacturing. We can easily manufacture hundreds of millions of cell doses. So we can easily do that. And in terms of shipping and to clinical sites and so on, it's much more easier. Being able to thaw these frozen cells and use them, whether it's for infusion and direct injection, etc., for in vivo studies, it's much easier. They will survive the thawing process and infusion process at a higher rate as compared to stem cells. So I think it makes them more usable or more friendly to use with some applications. Because think of it, if you're doing a clinical study that involves a few hundred patients and it will involve multiple injections or infusion of stem cells, you can imagine the task required to grow these stem cells, and not just grow them, but to make sure they're consistent, make sure that they're the same exact markers on all of them, cell markers, etc., that they haven't differentiated. It becomes very difficult. Whereas with fibroblasts, you have a much less worry in terms of differentiation, in terms of consistency and culturing.
Mm-hmm. Is there perhaps some, I don't, maybe disadvantage is too strong a word, but is there some challenge associated with fibroblasts as a therapeutic starting cell material? Is there some challenge that perhaps those other stem cell approaches don't face? Let me ask you this, right? I'll put it to you this way. I'm not a scientist. I'm not a scientist, but many of our listeners are. And if there's a potential disadvantage or challenge, solvable or otherwise, that is obvious to any one of those listeners, they're going to be sitting there going, Matt, why don't you ask him this, right?
Yeah.
So
no, that's a very good question. I think like any cell-based therapy approach, there will be advantages and disadvantages and limitations, right?
Mm-hmm.
So for fibroblasts, I'm sure there are limitations. That's why we're limiting their utility for testing to certain clinical applications that we think they would have an advantage. While there are quite a few other clinical applications that stem cells will be far significantly better. And we applaud that. I think we applaud their use because if the more clinical applications in utility that is approved, derived, or envisioned or invented by stem cells, we can certainly think of that as a possible use for fibroblasts as well. So limitations, yes, like anything, there will be limitations, and that's why we've limited our pipeline to certain clinical outcomes. And disadvantages, I think I would say the same thing. In terms of differentiating a stem cell into a tissue type, a certain tissue type might require only a certain amount of training to get them to differentiate. With fibroblasts, with certain tissue types, it might require more work to get them to differentiate. So I think that might be some of the disadvantages, but the disadvantages are based on clinical applications. I personally don't see a general limitation or disadvantage to using fibroblasts. I just see limitations, and I don't necessarily see that limitation as something bad. I think everything has a limit, and I think we have to operate within our limits and what we think these cells can do. And I think we have set a good boundaries for use of these cells currently.
Yeah. Oh, well said. It's good answer to that question. So I do want to talk about a little more specifically about some of those applications, and maybe. As I said, I think wound healing, I think, as far as your pipeline's concerned, the degenerative disc disease seem like obvious applications for a prolific structural grower. What about some of the other, the MS indication and the two oncological indications that you guys have identified, I think, so far in your preclinical program?
Well, for MS, our preclinical study, which we're submitting a manuscript, and I think we're also presenting it as a poster or a possible oral presentation at a meeting in June, it looks very promising. The preclinical study has shown that in an animal mouse model study, it's far superior in reducing the inflammation, reducing some of the signs of degradation that we see in multiple sclerosis. In our phase 1 study for human trial, which we carried out and completed in August of last year, late August of last year, shows a lot of promise. We found that it was a safety trial. We were basically infusing 100 million of these tolerogenic fibroblasts into MS patients, and we found that we not only saw them to be tolerable, they didn't show any adverse events. So it passed the safety aspect, but we were also monitoring some efficacy, and we saw some improvement in some of the tests that are typically done for MS patients, like, for example, the 25-foot walk test or the serial addition test. We saw some improvement there as well. So we're hopeful that once we are fully funded, we will submit an IND for a phase 2/3 trial to look at a long-term study with a greater number of patients and to look at the efficacy that we've seen as potential use for fibroblasts for MS.
first of all, I got to ask Pete, why'd you bring this guy on board as CSO, Pete? I mean, he doesn't have much to say. He doesn't even, does this guy bring anything?
He is.
I know I'm not much of a talker.
He's curious. He's curious. He's a sponge. He reads everything. I think I can send him a peer review paper at midnight, and he'll have it read by the morning and ready to talk about it. So I think he's a great partner.
Yeah, well done. Well done. In all seriousness, though, and I've got more questions about indications, but was it a strategic time for you last year to bring Hamid on and that role specifically, chief scientific officer?
It was, and as I told Hamid then, and continue to tell him, that is proof of us putting science first. He's employee number one, and that's why we wanted to put the science first. We weren't going to fill any other positions until we had that CSO position, and that made a statement to the market. It made a statement to our scientific advisory board. If you go look on our website and see, you're seeing international experts in the field of stem cells are on our scientific advisory board because they're excited about what we can do with fibroblasts. So, people say let the science lead the company. We're actually doing that.
Cool. Dr. Khoja, I was going to ask you a follow-up question on that MS indication. And I think I also read this, that, there's some evidence that your fibroblast therapies may have superior anti-inflammatory properties than some of the cell therapies that we're seeing or competitive or otherwise on the market.
Yes.
Do we know why that is and what those advantages might be? I mean, obviously inflammation is associated with any manner of disease. Anti-inflammatory properties equal good.
Absolutely, yeah. Immune modulation, right? Immune modulation is the key for some of these autoimmune disorders, and being able to control that and modulate the immune system to reduce the inflammation. For example, we find that the fibroblasts in our in vivo model reduces the expansion or prevents the expansion of pathogenic T cells. These are IL-17 excreting cells that is important in MS. It can reduce that significantly as compared to stem cells. But we also find that the fibroblasts can also increase the expansion of T regulatory cells, which are important in maintaining the homeostasis and controlling the inflammation for MS. And one of the things to note for our safety clinical trial, we found that within that time period, none of the patients went through an MS episode. So that was very intriguing to us, is to see if in a long-term study and that was, mind you, that was only a single infusion of 100 million cells. What can we do with multiple infusions during a time period of, let's say, a year? Mm-hmm. So we're really hopeful that we could manage to see some efficacious results. If you look at the main therapies for MS these days, there are either small molecules or antibody-based therapies, like they sequester B cells, etc., and they have long-term impact. They're very difficult to tolerate. The side effects are very bad comparatively. So I think what we hope is that with cells, we would be able to reduce a lot of these major side effects and without having to deplete a certain population of cells in a human body, which could impact other immune issues later on in life.
Mm-hmm. Okay, it's I know it's real early days in your preclinical cancer activity, but do you have? I mean, is there anything that can be said there in terms of what your approach might be or your hoped mechanism of action?
Yeah, and in terms of we're looking into the mechanism of action, but in terms of just some of our initial preclinical study, we find that we've been able to reduce tumor size by direct injection of fibroblasts in the tumor microenvironment. These are human dermal fibroblasts. So we've seen some reduction in tumor size that has been significant as compared to control. we're going to look into more into what that mechanism of action might be and which type of cancers exactly that would have the most efficacious results. It's at the beginning of our study. We're hoping that I could expand the study for cancer perhaps in the mid second or third quarter of next year. But for now, I think we are focused on multiple sclerosis. And right now, I think one of the first studies that we are doing now for wound healing for animal model, I think that's going to be important, because one of the things is that for multiple sclerosis and for degenerative disc, you have to look at long-term at the patients, right? Typically a year, 2 years. Whereas with wound healing, typically when we're looking at diabetic foot ulcers, etc., which is predominant, and we should be able to see results within weeks rather than months and months. So I think that's why we're pushing ahead on wound healing at the moment, but we will certainly pick up cancer and hopefully by second half of next year.
Yeah. Okay.
You know, Matt, the promise of cell therapy and all that Hamid's talking about is actual cures. And I think you'll see that gene therapy, immunotherapy, you look at the last couple of Nobel Prizes, one was in CRISPR and one was in immunotherapy for Dr. Allison here in Houston, MD Anderson. And so what the promise of cell therapy, gene therapy, and immunotherapy is that you actually have a cure for these chronic conditions. And we've been, I took pharmaceuticals this morning to control cholesterol and high blood pressure. And that's what we have available to us. But we can't find an example in the last 60 plus years of big pharma where they've actually cured a chronic disease using chemicals. It just doesn't really work.
Yeah.
There hasn't been one. And so the promise of an actual cure for a chronic disease we've been working on for 60 years at the chemical level hasn't happened. It's going to happen through cell therapy. It's almost, without sounding too granola or hippie, it's going to be by teaching the body with its own cells to cure itself. I mean, the disease emanated from that body, and to use an external chemical compound to cure that disease almost doesn't even make sense. Mm-hmm. So I think that's where the growth of medicine over the next couple of decades will be centered on cell therapy. And I think you'll see a first couple of commercial applications where cell therapy will be approved this year for chronic condition. And I think that'll kick off the race will be on at that point.
Yeah, that's it's absolutely. I mean, excellent points. And a good segue to another element of the conversation I wanted to have with you too. And that's, I want to get your perspectives on, here you are developing, you're working on developing a cell therapy that is derived from donor cells, i.e. harvested from, excess surgical material. And you're taking that, you're attempting to create an allogeneic, off the shelf application for just about anybody. And then I look at like Dr. Khoja's background and I go, oh, this guy worked in like genomics, right? Like he spent quite a bit of time on that. And I talked to any number of other, founders of so-called, personalized or precision medicine companies who are working in, just as much earnest as you guys are to develop specific medicines to address specifically Pete O'Heron's cholesterol and high blood pressure, not mine, not Dr. So, right. So what I see here in my simple-minded kind of way is some conflict. I see some conflict. Like we're hearing from one side of the market that the future in biologics is in highly precise and personalized medicine. And then we turn around and we go, oh, well, there's a company that's looking to make medicine for everybody to heal their wounds, created from cells that, we don't even know who those cells came from, but they're allogeneic and they're going to work in all of us. Help me reconcile the conflict here.
Right. I mean, genomics, epigenomics, they're all tied in to cell therapy, right? I mean, they have to be. It's a matter of how things work. I think the more we have, the better understanding we have of genomics and epigenomics, the better we will understand tissue regeneration, tissue-based or cell-based therapies. Because if you think of it, for example, fibroblasts are not genetically different than any other cells in your body. They're epigenetically different. That's what makes them fibroblasts. It's how epigenetically different they are as compared to the other cells. That's what differentiates one cell type from another. And I think, and the better understanding we have of epigenomics, the better we will understand on how to destine the fate of some of these cells to become certain cell types. So epigenetic studies, epigenetic-based experimentation and studies and markers, that's something that I'm certainly looking at for some of our in-house research, because that's an important aspect of being able to carry out some of these tissue regeneration and cell therapy. I think it's important. Even if you look at the fibroblasts, there are many multiple subtypes of fibroblasts. Genetically, they're identical, but epigenetically, just certain genes being on and off in one as compared to the other, that's what makes them different and destined to be their fate on becoming a different cell type is completely different. So I think I don't see a conflict per se between genomics, epigenomics, and cell therapy. I think they go hand in hand, and they have to. I just don't see cell therapy being successful without the knowledge that has been obtained in epigenomics. Yeah. And I think that's one thing that we don't focus on a lot is epigenomics. It's I think, in my opinion, while genomics is very important, epigenomics is going to be the next phase in a lot of cell-based therapies and cures for human disease.
Mm-hmm. Okay. What does that mean from a practical application standpoint? So let's say there's a situation where, perhaps, I don't want to make any assumptions about the market potential for the candidates you're working on, but perhaps, wound healing it's an unlimited market. I mean, it's an unlimited application in that space. So based on what you described to me, would it be incumbent upon FibroBiologics to create like several variations of a wound healing therapeutic to fit, to fit specific people or specific?
Matt, it's interesting. We actually filed intellectual property on that about 4 years ago, where we grade the donors and we grade the recipients. And we see a day in the future where you'll have 3 to 4 different grades of donors, 3 or 4 different grades of recipients. And depending upon the disease path, you'll match up the donor with the recipient. That's really looking over the horizon on this, but that's where the future is. So to the extent that you can actually target, I think we can, what would you call that, Hamid? Optimize the donor to the recipient. I think you'll see that in the next phase.
Right. I mean, and this might be a good way to segue into personal medicine, right? Mm-hmm. You could easily take a dermal punch from a patient, isolate the specific fibroblast necessary. Let's just say these patients have diabetic ulcers. You can easily take cells from them and grow it into the fibroblasts that are necessary for their wound healing. It just takes a few weeks. So that's a possibility. But we also find that fibroblasts, like stem cells, have minimal immunogenicity, and that they're tolerable. And we've seen that even in our clinical trial for both degenerative disc and multiple sclerosis, where we infused 100 million cells, we didn't see any adverse events occurring short-term and long-term. And the same with the degenerative disc study with 21 patients. We didn't see any. So I think while we can certainly consider them tolerogenic and being able to use the tissue from different patients, as Pete mentioned, grade the patients immunologically to several different groups and use them. Certainly, that's a possibility. And I think we should be able to also determine, let's just say, a certain subpopulation of fibroblasts will work well with diabetic ulcers, but some other ones might work better with pressure or friction based ulcers such as bed sores, etc. So I think we're keeping an open mind in terms of looking at all those possibilities and potential for our fibroblasts.
Very good. And Pete, thanks for jumping in there and answering my question before it was fully formed, because I wasn't sure how to finish it, and I didn't know if it was really smart or really simple. But you made me sound really smart when you said I was looking way over the horizon. So good.
Yeah, there you are.
No, great question so far.
Oh, it's good stuff. Yeah. I want to give you an opportunity to give us an update on your. We're actually running pretty short on time here. I feel like I could continue asking you guys questions that go around in circles here all afternoon, but we're running short on time, so I'm going to need to wrap things up a little bit. But I want to make sure that I give you an opportunity to discuss, what's next, what's going on in the pipeline, with the, whether you have any big milestones kind of coming up in the next 6 months to a year. Give us an indication as to where things are going.
From a business standpoint, we're on kind of an IPO path. So we'll be taking the company public either through a reverse merger or through a SPAC or through a traditional IPO. So I think you'll see that ramping up of resources to pursue even more clinical pathways. So I think that from a business aspect, that's the next big thing. And then I guess I would add to that Hamid will be getting his own lab and his own manufacturing facility to go with that, which is the dream of every CSO. He'll be able to leave his office and walk straight into his lab. So
I'm gonna go
that's
ahead
exciting.
I'm going to go ahead and assume this is the first time that Hamid's heard that. You were saving this episode of the Business of Biotech. Great news.
No, Pete and I have a good, transparent relationship. So he's.
Awesome.
I've been aware.
Was it Pete? Was it the intention all along to go the IPO route, or have you sort of juked and jived a little bit as you've grown and matured the business over the years? What was sort of the intention there?
Well, we were a private company for a long time, and I think the intention has always been to either take some of these disease paths and license them out to major pharma, or to what we've done is we've pulled the crown jewels out to form FibroBiologics for the purpose of taking that public. So, if you want to get to the next level and you want multiple clinical programs going on, you absolutely need to have access to those resources, and you can get that through the public market. And I think they'll be receptive to this. We are, again, everything we're attacking. First of all, it's the first time anyone's ever done it. So the IV infusion of fibroblasts from allogeneic fibroblasts had never been done before. And we're attacking diseases that have no cures. Everything that we're working on, there are currently no cures. So from that standpoint, I think it'd be attractive for the public markets.
Yeah. Very good. Well, hopefully your timing is good. Yeah. I don't know if there's ever like a bad time per se, but the way that the biotech markets have looked for the past, 6, 9, going on 12 months, newly IPO'd companies have been taking quite a beating. Neither here.
I hear from my surgeons on a daily basis, the market's down. What are our plans? The market's up. We got to go. And I said, you know what? You go when you go, and it's impossible to time the market. So you just hope that the day that you go out is a good day in the market, but you really don't have control over that.
Yeah. Good stuff. So any other kind of concluding thoughts on the direction of FibroBiologics and what's coming up next?
Right now we're building out the rest of our management team, moving into our new space. And I think you'll be seeing multiple announcements coming out. We just filed some really, I would say it's historic IP with respect to the immune system that we'll be announcing in the next few months. It's really, really exciting. Mm-hmm. An extension of human life type of excitement. So wow. We'll be announcing that, and not to put too much pressure on Hamid, but he'll have
not at all.
He has a lot to work on.
He's got that shiny new lab.
Good for work.
Got that shiny new lab to work on it in.
So. Right.
Awesome. Well, listen, I know there's more to talk about. There's just not enough time right now, so we're going to, we'll stay in touch and maybe do a part 2 here maybe when that paper comes out, when there's a bit more to talk about and the company
Sounds great.
moves a little further down the road. But it's a great start to the conversation because I learned a lot. I appreciate you both for subjecting yourselves to my sometimes naive questions and offering the education. I think it was valuable to me and certainly valuable to our audience. So thank you guys for coming on.
Thank you
Great.
for having us.
Thanks for having us. Appreciate it.
So that's FibroBiologics founder, chairman, and CEO Pete O'Hearn and chief scientific officer Dr. Hamid Khoja. I'm Matt Pillar, and this is the Business of Biotech. We're supported by Cytiva, which demonstrates its commitment to early stage biopharma at the Emerging Biotech Accelerator. You can find at cytiva.com/emerging biotech. Visit Cytiva, then visit me at bioprocessonline.com, where I invite you to subscribe to my newsletter, learn from the writing of your peers, and listen to the entire library of Business of Biotech episodes. If you're enjoying conversations like these, please subscribe on Apple, Spotify, or wherever you listen to podcasts and leave us a review. See you next week. And as always, thanks for listening.